Hello, Dr. Chen. This is our presentation, MSH2 gene mutations and its effects on hereditary non-polyposis colorectal cancer, HNPCC. My name is Jordan Branch.
I'm Lady Wadike.
And I am Tier Lacey.
So HNPCC, hereditary non-polyposis colorectal cancer, is the most common form of hereditary colorectal cancer. It's inherited as an autosomal dominant syndrome and is the result of a defective mismatch repair protein. It accounts for 2 to 5% of all colorectal carcinomas and about 90% of all colorectal cancers in HNPCC patients demonstrate a high microsatellite instability. HNPCC is associated with other cancers such as gastrointestinal cancer and cancers of the reproductive system, and the increased cancer risk is due to an inherited mutation that degrades the self-repair capability of DNA. Symptoms of HNPCC include blood in the stool, extended periods of constipation, changes in bowel habits, weight loss for unknown reasons, and diarrhea that is not related to your diet or illness.
Okay, so the molecular effect of HNPCC, as we said before, it is an inherited mutation in one of the DNA mismatch repair or MMR genes, and it appears to be a crucial factor. MMR genes usually make proteins that identify and correct sequence mismatches that may occur when the DNA is replicating. In HNPCC, the mutation inactivates the MMR gene, and that leads to the accumulation of cell mutations, and that greatly increases the chance of malignant transformation and cancer. Researchers have identified 7 distinct MMR genes, including MLH1, PMS1, MSH2, and MSH6, and we will be specifically talking about MSH2. MSH2 is a member of a set of MMR genes that provides the instructions for making proteins that play essential roles in DNA repair. It fixes mistakes that are made when the DNA is copied during replication in preparation for cell division. It joins with 2 other proteins, MSH6 and MSH3, MSH6 and MSH3 each being produced from a different gene, and all 3 of them make a protein complex, and that identifies the locations on the DNA where mistakes have been made when the DNA was replicating. Another group of proteins, MLH1 and PMS2, form a complex that then repairs the errors. And this slide is showing the location of the MSH2. This table basically summarizes the relationship between MMR genes and the frequency of HNPCC cases. And as we see, MSH2 is responsible for about 45 to 50% of these cases.
And now we will start with our MSH2 mismatch repair part of our lab. So our experimental objective was to gain a deeper understanding of the effect that a MSH2 mutation has on tumor suppression, which may lead to the development of HNPCC in humans. So our hypothesis is that the presence of an MSH2 mutation will encourage cancer formation. The purpose, overall purpose, is to identify which mutations affect MSH2's repair function. So in part 1, we isolated the plasmid DNAs from the 3 E. coli strands, and then 2, we measured the concentration and quality of our DNA samples using the nanodrop instrument. So part 2, we digested our 3 plasmid DNAs using restriction enzymes XBA1 and APAL1. And then secondarily, we ran the digested products on agarose gel to determine which plasmid DNA contains the MSH2 insert. And then in part 3, finally, we amplified the region of the MSH2 gene that contains the mutation. So our methods. So part 1, we did the plasmid mini prep, and we measured the DNA concentration, and we used the 260/280 ratio using nanodrop 1000 spectrometer. Then part 2, day 1 of the restriction enzyme plasmid DNA, we thawed all the reagents, we diluted and made different dilution samples. We diluted the 12 microliter plasmid DNA in 100 nanograms per microliter, and we repeated this step for the 3 plasmid DNAs. And then we also added buffer and the percentages of the restriction enzymes and added that to the plasmid DNA. And then we also added 20 microliters of control reactions. So we did our 3 controls, and then we added fast digest buffer and then plasmid DNA. And then we incubated the reaction at 37 degrees Celsius for 30 minutes. And we had 6 samples total: the SPO cut, SPO uncut, SP1 cut, SP1 uncut, SP4 cut, SP4 uncut. So part 2, day 2 of the restriction enzymes of plasmid DNA. We did the poured the melted 1% agarose gel in the buffer into the gel tray, and then we let the gel solidify, made the gel, and then performed gel electrophoresis and in the 3 lanes. So we had 5 microliters of 1 kb DNA ladder in lane 1, and then 10 microliters of digested products in the 3 lanes, and then 10 microliters of undigested products and another set of 3 lanes. So for a total of 7 lanes. And then after that we did electrophoresis and then imaged the agarose gel. And then part 3 we did PCR. So our PCR protocol we used was 95 degrees for 5 minutes, 95 degrees for 1 minute, 55 degrees for 1 minute, 72 degrees Celsius for 1 minute, and then return to step 2 for as many times as necessary. And then we did 72 degrees Celsius for 10 minutes and 4 degrees Celsius. It was our hold temperature. And then part 4, we did restriction digestion of our PCR product. So day 1, we thawed reagents, and we did 3 20 microliter digestion reactions containing the buffer, the restriction enzyme, and PCR product. And then we did the same thing for our control. We had 3 controls of undigested product, and that contained buffer and then PCR product. And then we incubated this, and then in day 2, we poured the melted gel, the buffer, and let it solidify, and then prepared the samples. And we added loading dye and undigested products, and then we mixed it and spinned it down briefly, and then performed a gel electrophoresis on the samples. So the first sample, the 5 microliter and 100 base pair plus DNA ladder, and then the second one was the 10 microliter digested products in 3 lanes, and then third we had the 10 microliter undigested products in another 3 lanes. And then we did gel electrophoresis and then imaged the agarose gel. And then part 5, the part 1 of the yeast formation. So basically, our formation reaction that we used was plus the plasmid, the transformed plasmid. So that was 0.5 microliters of yeast cells in plate solution. And then we had 20 microliters of 1 molar DDT, then 5 microliters of denatured carrier DNA, and then 10 microliters of plasmid DNA. And then in our control plasmid, we had 0.5 microliters of yeast in plate solution, 20 microliters of 1 molar DDT, and then 5 microliters of denatured carrier DNA, and then 10 microliters of water. And then part 2, we mix the contents of each tube, carefully pipetting up and down, and then we incubated the tubes for 40 minutes at 30 degrees Celsius. Then we transformed them using a heat shock technique for 15 minutes and 42 degrees Celsius in the water bath, and then we centrifuged, and then removed the supernatant, and then resuspended the cells in sterile water. And then we transferred the entire volume into SD negative His and trip and ultra plate, and then spread it evenly across the plate using the sterile glass beads. And then we followed the A through E procedure, and then after that we incubated the transformation plates upside down for 24 to 48 hours in 30-degree Celsius incubator, and then colonies should have appeared in 2 days. And we recorded the number of colonies on both plates. And then part 6 was our FOA assay. So as you can see here, the picture on the left, it had the square that we drew on our plate. So we can determine, well, identify what each colony that grew or did not grow was. And then for flow cytometry, on day 1, we arrested the DNA replication using the hydroxyurea, HU. We had 2 tubes, plus HU and then minus HU. And we added 1 microliter of log phase yeast culture per tube. And then we centrifuged for 1 minute, discarded the supernatant, then resuspended in the SD medium, and added the 50 microliters of 2 molar HU to the plus HU tube, and then added sterile water into the HU negative tube, as well as incubated at room temperature for 1.5 hours. And then we also did wet mounting and suspended the yeast cells. And then under the 40x objective, using a cell phone placed on the cell phone microscope. So we took an image of that using the cell phone, and we centrifuged both samples, the HU plus and HU minus, for 1 minute, discarded the supernatant, resuspended in sterile water, centrifuged again for 1 minute, discarded the supernatant, resuspended in sterile water, and then stored the cells at 4 degrees Celsius. And then we did flow cytometry. Day 1, we reactivated the cell division and fixed the cells. And day 3, we did data analysis. So now results.
So these are the results of all of those labs that she just described. The first one being the plasmid mini prep. So this chart just shows the original concentrations of our samples and the dilutions that we made using water in a volume of the plasmid to dilute the samples to a concentration that we could use for the experiment. We did not dilute SP4 anymore because the concentration was already lower than 100. This slide shows the gel electrophoresis results for the first gel electrophoresis that we did to figure out which sample had the mutation. It showed that SP4 is the sample with mutation in its sequence. The presence of the double bands in the results show this. By showing that there's an extra enzyme restriction site, whereas in SP0 and SP1 it is only cut once. This gel electrophoresis is to show the use of the restriction enzyme to distinguish the mutated SP4 from the original sample, the wild type sample. So as you can see, row 4, which is the SP4 cut, is the only row that has 2 bands, which shows that it has that extra restriction enzyme site, like I said before. And this is what when we use NEB cutter to figure out which enzyme that we could use to distinguish the wild type, our mutant SP4 from the wild type. So we figured out that we could use BST-Y1 because that's the enzyme that cuts somewhere on the mutant, but does not cut in the same area on the wild type. This shows the sizes of the samples after cutting them with that restriction enzyme. And this shows the APE alignment that we did, comparing the wild type on the top with the mutant on the bottom. So you can see where the red marks are, that there's 2 points of mutation at the points 5487 and 5488 between the wild type and the mutant. Next, we did the yeast transformation lab. The left picture, which is blank, shows a growth plate that's lacking histidine and tryptophan, which is necessary for the yeast growth, which is why you don't see any growth on the plate. The right shows the mutated MSH2 transformed with PSH44, which incorporates the histidine and tryptophan markers, so it allows the yeast to grow, which is why you see those colonies grown on the plate after that transformation. And this is our FOA assay data. So this is when we made the grid on the plate that Tiara described earlier, which shows which sample it is and the concentration. And for this data, there's a low colony count in the 100X wild type section, but there's no conclusive evidence that shows that this is a result of frame shift repair by MSH2 protein. The initial cell count is uncontrolled, and this chart on the bottom just kind of explains that in more detail. And then this is what the results of the flow cytometry lab would somewhat look like if we were to complete it using our SP4 mutant in the wild type. So flow cytometry is completed to be a method of counting the amount of cells on the plate. So these humps, I guess you could call them, would be a cell count. So if we were to do this with the SP4, we could use these to determine if that mutation affects the cell cycle and either lessens or increases the number of cells that grow in the presence of that mutation.
Implications in the human MSH2 mutation. So out of our interventions, so the mutations in the human MS2 here, and the mutations in the yeast MSH2 led to functional studies. So we did the overexpression, and we were determining whether the mutation was functional.
Okay, so in our discussions and our conclusions, when we used APE to align the wild type PMSH2 and the possibly mutated SP4 and clustal omega to align those translated protein sequences, we have proof that there is a missense mutation at point 704. At that point, glycine in the wild type is changed to serine in the mutated sequence. So it's a G704S mutation. APE and NEB cutter analysis suggested that the BST-L1 enzyme, our restriction enzyme, differentiated between our mutant and our wild type. So this suggestion comes from the presence of the BST1 restriction site in the mutated sequence of SP4, but not in the wild type sequence, which is how we were able to tell them apart. This all leads us to human MSH2 requires some sort of ATP or energy in order to function. Future directions for this study would be necessary to find out if the lack of ATP in the mismatch repair could affect the actual repair. And also what effect the MSH2, MSH6 complex has on the mismatch repair, and what would be the consequences of a mutation in that complex other than MSH2. Relevant research would include this paper which we found, which is evaluating the cancer risk in MLH1, MSH2, and MSH6. This is relevant because they are researching, furthering what research we did in class, especially with MSH2. The purpose of that research was to look at the summative lifetime risk of LS-related cancer in similar MMR mutations, which would include MSH2 mutations, and to see the cause that seems causative in distinguishing cancer risk profiles. Also, female MSH6 mutation carriers have a low CRC risk. So basically, the study evaluated 246 individuals, and it determined the cumulative lifetime risk of developing cancer of these individuals. They found that the risks of cancer at age 70 years of all MMR genes or MMR mutation carriers was similar. And they also found that MSH6 mutation carriers at the age of 70 years old have a high risk in developing CRC. And it was lowest in both males and females when compared to carriers of MLH1 and MSH2 mutations. These are the study population characteristics. It breaks down how many families, mutation carriers, and males and females of each MMR mutation was included in the study. And then this is the cumulative risk in female mutation carriers, and it increases as years increase, and it's the same thing with males. And so the conclusions of the study basically said that each mutated MMR gene had a distinguishable cancer risk profile, and also that female MSH6 mutation carriers have a low CRC risk and a higher risk for developing endometrial carcinomas. Future directions of that particular study would be to continue to study the effects of missense mutations of MMR genes and focus on their causations and regulation processes so that we can conceive ways to decrease mutation rates and thus lower the risk of cancer forming. These member of our class for sharing their pictures and information in terms of their labs with us as well.
That's all, folks.
Why are you laughing? I stopped it already.
Very random.
What the hell?
Someone who wasn't even.
Hold on a second.
Wait, is it still?
Yeah,
it's still recording?
No, it's not.
Oh,
Wait.
someone.
It is because it stopped recording.